So, what I'm going to be talking about is how you actually handle the data that comes off of the machine once you've gone through all of the processes, mainly in the wet lab that Anu has told you about to actually generate the data and run the sequencer in the first place. So my little summary slide is a little bit different from Anu's. I'm essentially taking over from the final arrow that she had along the top of the screen for data analysis. And there's lots of different ways that you might divide up the data analysis pipeline. But the way that Illumina like to do it is to talk about primary, secondary, and tertiary analysis. So primary analysis basically means it's sort of overlapping a lot with what Anu said, actually, but essentially what goes on inside the machine, assessing the quality of each read as it comes off the machine, identifying the fluorescent signals that are coming from the nucleotides as they're added onto the growing nucleotide string, and turning that from sort of fluorescent pictures into hard data, AGCTs. So as far as primary analysis goes, I'm going to talk a little bit about how this is encoded in, and stored as a quality score. A little bit more about base space and how you can use base space after the run to assess the quality of that run. And also about how you can get the data out of base space or off the sequencer and into a format that's useful for further analysis later on. Under secondary analysis, I'm going to stop around about where this dotted line is because I don't really have time to go into everything. But the first stage is essentially checking the quality of your data, making sure that it represents what you want it to and that it's suitable for use in further analysis later on. So I'm going to talk about a couple of common tools for that, FastQC and Trimmomatic, which I'll go into a little bit later. If I had a bit more time, I'd then talk about aligning or assembling the reads, so going from little short sequences of a few hundred base pairs into long chains of kilobases of DNA, and also comparing that to perhaps a reference genome that's already been sequenced and identifying differences between that. A lot of this will be spoken about a lot later on. We'll go through the workshops and demonstrations, so you'll actually have a chance to do a lot of this stuff on your own later on. Tertiary analysis, I'm definitely not going to talk about that because that's a lot further down the line, but that's more your sort of aggregating all the data from lots of different samples together, comparing those, perhaps identifying an entire population and comparing different populations with each other, or the really sort of big scale analysis that really links the sequence data into what's the biologically relevant outcome, and really answering the question that you sort of wanted to answer in the first place. So, just to backtrack a little bit, we've talked quite a lot about cluster generation in the MiSeq. So you have your flow cell fits in here, and on your flow cell are all the little sequences that are your candidate sequences combined to in order for the bridge amplification to take place. As you've seen, this is roughly what the sequencer sees, all these little fluorescent dots for each read. The software interprets that as different colored dots that correspond to each of the 4 bases. But the first problem that we have is that each of these spots is going to have a slightly different confidence associated with it in terms of how happy you are in identifying, say, this spot as a green is quite nice because it's separated from all of the others and you can be quite sure. But this one here, you're not sure whether it's purple or orange because there's a slight overlapping region. So you can probably say with a fair amount of confidence, this is a purple dot, this is an orange dot. But in the middle bit, you've got some area where you might get a little bit confused. And I'm not sure if you can see this one very well, but this dot here is actually a slightly different color from all of the others. We're not quite sure. It looks slightly greenish perhaps. Maybe it's got a little bit of purple in it. We're not quite sure what color to assign that one. And so the machine looks at all of this. It has algorithms that decide which base to assign to each of the different clusters. And it also looks at clusters that it's not very sure about and filters out some of the ones that have very low quality. And so you end up with your raw data that has all of the clusters in, and also some filtered data which has a sort of a subset of the data that's already had a little bit of quality control applied to it. So you need a way of codifying the quality scores that are coming off the machine. How confident is the machine in each of the bases that it's assigned? And what Illumina use, and actually is kind of a universal standard now, is a system called Phred, which originated during the sequencing of the human genome and came out of the first program that produced highly accurate base scores. And this program happened to be called Phred. The program itself is not really used so much anymore, but the actual system that it used for quantifying the base call accuracy is very widely used and is therefore called Phred scores or sometimes Q scores. So it's a fairly simple relationship between the quality score and the probability of having an error in each of the base calls. So for example, if you know that your quality score is 30, then the minus 30 divided by 10 is minus 3. 10 to the power minus 3 is 0.001, which is an error rate of 1 in 1,000. So a general rule of thumb that Anu sort of slightly touched on is that generally anything with a Q score greater than 30 is acceptably high quality, and anything lower than that is often considered to be too poor quality to continue with. It depends a little bit on exactly what analysis you're doing, but that's the general rule that people go for. So the final thing that the machine will do for you after calling all of the bases, you then have a big pool of lots of different sequences. And as we said, each of these sequences has these adapters at the end, including the index sequences. And so it reads the index sequences, cross-references that with the sample sheet, and says, okay, so sample 1 should have this index, so these sequences are all coming from sample 1, these sequences are all coming from sample 2, etc., and separates it all out for you into the individual samples. Then, as we've said, while the sequencing run is going on in real time, all of this data is being uploaded into BaseSpace for you. So you can check some of the parameters while the run is going on, and you get more data at the end of the run once it's able to do all of the analysis together. So if you were to open up BaseSpace immediately after a run, you'd see something like this as a summary of the run overall. This gives you some of the details, tells you the samples that you've got, and also a few of the parameters that I'll go into in later slides in more detail. So let's just work along some of these tabs along the top to see the sort of data that you get out. So if you go into the charts, you get a representation of the flow cell itself. You can look at various different parameters, but the sort of default that we're looking at the moment is the intensity, so the fluorescence that's seen, and it's divided the flow cell up into individual units as well. So for this unit, you can see that down the left-hand side, you're getting more fluorescence intensity coming out, particularly than the bottom left. And so it shows that the cluster generation across the whole flow cell is not completely uniform. You get some areas with more clusters and some areas with less. And if you have problems in your run, you might be able to identify that there was a particular problem in one area of your flow cell from diagrams like this. The same data can be represented in a line graph like this. So this shows the average fluorescence intensity across the whole flow cell per cycle of the sequencing run. And you generally expect a shape like this where you get slightly higher intensity towards the end of the runs as more clusters generated and slightly more sort of fluorescence comes out from having slightly more molecules on the flow cell. I think Anu's already spoken about these heat maps and the Q-score distribution. I'll talk a little, now that you know what Q-scores are, you can see that the green bars represent all of the scores that are above Q30, so they're applying the rule of thumb that I mentioned before, and very few of the scores fall below 30, so this indicates that it's a good run with high quality. If we look at some of the graphs that you get in more detail, this is quite a useful one showing how many clusters are formed on the flow cell itself. And as I mentioned, it applies some filtering as well. So this blue box chart here, you can't see all that well that it's a box chart because it's quite compressed. But the blue one represents the raw reads from the total flow cell, and the green box represents all the data after the filtering has been applied. So the red line shows the median cluster density. The box itself represents the 50th percentile, so half of the data falls inside the box, and then the whiskers coming out at the top of the bottom of the box represent the range of the data from right at the top to right at the bottom. The ideal cluster density, or raw cluster density, should be between about 120 and 140 on this graph. So this shows it's very slightly over the ideal. And you can see that there's a fairly large gap between the blue box and the green box, showing that quite a lot of the sequences have been filtered out. And so this probably indicates that perhaps the run had slightly too many clusters, so quite a few of the dots will have been overlapping, and the machine won't have been quite so confident in how to score a lot of the nucleotides. There's also a useful table that shows you a lot of the metrics for the run, so you can see exactly how much data you're getting out, how many reads has taken place in the whole run. And if you were to look at this during the run itself, it actually gives you a prediction of how much data will come based on how much it's seen already. So that can be quite useful just to sort of make you feel confident as it's going on that you're going to get some good data out at the end. The indexing QC is also useful, so once it's finished the whole run, it assigns the individual reads back to the samples, and you then get a graph like this that shows you what percentage of each sample, what percentage of the total reads in that run were assigned to each of the samples. So usually you'd expect each sample to have roughly an equal amount. This isn't too bad, although there's a few you can see. This one has perhaps a little bit too much representation, and a couple of them don't seem to have very much at all. So you might find that you'd need to repeat these samples with not very much data in them. And you can also review your sample sheet, and if you happen to have made a mistake, I think you can reload a new sample sheet to sort of reanalyze the data based on that as well. So that gives you a rough overview of what you can do in base space. There's a lot more other stuff that I don't have time to go into now, but when you do your own sequencing, you can play around in base space, see what kind of information they have. And Illumina have very good documentation around it all that tells you exactly what you're looking for and how to identify whether it's good or bad data as well. But now we want to get the data off the machine and actually start using it and finding out something interesting from it. So our next problem is Illumina like to use this file format called BCL, and they use this across all of their machines. Essentially, you get one one file generated for every cycle of the machine, and it stores all of the base calls that the machine's made before the samples have been demultiplexed. We then need to convert from this BCL file into a FastQ file, which is a more generally applicable format that contains all of the sequence data now separated into each individual sequence read. So it's been demultiplexed, and it also comes along with all of the quality scores for each of the for each of the base calls as well. So if you've got your machine linked up to base space, as you do here, then this conversion from BCL to FastQ will happen automatically through base space. But if you take your data directly from the machine onto your own computer, then you need to use separate software to do this conversion. But the FastQ files themselves look a little bit like this. So you have 4 lines for every read that is in your file. The first line is a sequence ID. So it always starts with an @ sign, and then has some, in this case it'll be an automatically generated sort of fairly long string that the machine assigns to each read. The second line is then the sequence data itself. So this is a relatively short amount of sequence, but it could continue way off over here if it's a really long sequence. You then have a third line which always has a plus in it here, and you can repeat the sequence ID just to sort of double check that you've got the sequence data and the quality data lined up together. And then finally the fourth line looks like complete gobbledygook to us, but the computer understands this as quality scores. So each of these characters represents a particular score on the Phred scale that I introduced before, ranging from the exclamation mark, which is extremely low quality data, right up to I think that's called a tilde, isn't it? Which represents extremely high quality data, and ideally, I don't know, 30 is sort of around about here or something as your cutoff point. So that takes us roughly to the end of the primary analysis sequence, and as you've seen, a lot of that happens automatically, so you don't need to worry about it too much, but you do need to understand what's happening inside the machine and how it's converting from one sequence, one data format into another. If we then move on to secondary analysis, the first thing we want to do is to make sure that the data we're getting out is actually of a reasonable quality. So for this we use, there's actually a lot of different programs you can use for this, but a very common one is called FastQC. And it was specifically designed to identify problems that are caused either on the sequencer itself or further back in the pipeline when you're actually preparing the sample and doing the library preparation. It looks for biases in the data, so it starts with the assumption that all of your data is going to be well balanced. For example, you'll have no particular bias towards any particular base. And then looks to see whether there's any differences from that assumption. You can run it either in standalone mode, which is like an application through your web browser, in which you upload the data, run the analysis, and then you can see it almost as a web page with hyperlinks that you can click on to view each of the different analyses that it does. This is good for running a few samples, but if you want to run lots of samples altogether, it's much easier to run in non-interactive mode, which means a program that runs through the command line. You type in commands into the computer, and rather than getting pictures up on the screen, you just sort of get text information back. But then later on, you will get a report file generated that you can then save and view later anytime you like. So before we have a little look at the data that you get out of it, it's worth just noting, as I said before, FastQ assumes your data is going to be random and diverse. And depending on your experiment, this might not be the case. So you're very likely actually to get some sort of warning or failure in at least one of the analyses that you do. It doesn't necessarily mean that your data is no good. It might actually be exactly what you expect, but it's not what FastQC expects. So it might sort of give you a false warning occasionally. Of course, sometimes it'll warn you for good reason. And on the other hand, if you get a pass, it doesn't necessarily mean your data is good. So you really need to understand a little bit about what it's looking for and how it makes a decision as to give a warning or a pass. And they give very good documentation as well about how all of this works. So I'll put up a link a little bit later where you can find that. If I just get out of here for a second. I'm just going to show you some examples of the reports that you can get. Okay. So on the left of the screen is an example of the data that you might get from a very good sequencing run. Actually, in this case, all of the data is passed. On the right is a bad sequencing run that hasn't given you very good quality data. So we start off, it's a bit small to see, but you get some very basic statistics about the samples that you've put through. So you can double check that you're actually analyzing the right samples. It tells you things like the sequence length that you've identified, the percentage of please see, things like that. As we work down, you then have the per base quality scores. So along the bottom of the graph is each of the bases, each of the reads that has happened in during your sequencing run, and then the average quality score that's been assigned across all of the samples to the first, the second, the third base, etc. So in the good quality score, all of it falls in this green region, which again is using the rule of thumb that a score above 30 is good. But in the bad quality data, it starts off quite nicely actually, but towards the end of the run you get huge variation in the data and much worse quality scores, so you're going to start worrying that the base calls towards the end of your run are not going to be accurate. It then has a look at bad data here. Actually looks a lot more interesting than the good data. So what you want to see is just this big blue box. This is quite similar to the heat map that I showed you in base space, where it looks at the whole flow cell and breaks it up into individual sections. And if you get variation from the average, then it'll show up in this heat map here as regions of sort of high or different colors. So if you get small regions like this with a little bit of variation, you don't need to worry too much. But if you see large amounts of consistent variation, say here, then it might indicate that that particular area of the flow cell might have problems. Maybe had a little bubble was introduced or something like that. Another demonstration of the quality scores. It's not always looking at quality scores, but in this case, it's looking at the data sort of flipped around the other way. So rather than looking at each individual base call, you now have the quality score along the bottom and the number of reads on the y-axis that have an average quality score of 30, 32, whatever. So in the good quality data, you can see almost all of the scores are well above 30, whereas in the bad quality data, you still have quite a few that are around high 20s, low 30s, but there's this sort of subpopulation of sequences that have very low quality scores. So maybe later on you could filter out these bad ones and just keep the good quality ones. We then move on and look at the sequence content per base. So as I said, it assumes that all of your samples are going to be random and diverse. So you'd expect roughly similar proportions of A, G, C, and T bases. So each of the 4 coloured lines here represents each of the 4 bases. And if you get a flat line where all of the bases seem to be roughly equally distributed, then that's good. If you start getting a lot of variation in it, wobbling up and down like this, that indicates perhaps there's some bias in your data. That's then represented again in a slightly different way. So we knew the overall GC content of your sample was about 45% in this case. It applies a theoretical distribution, so it assumes that you have sort of a normal distribution around that average. And then in the red line shows the actual distribution. So if it closely mirrors the blue line, that's good. If you get these sort of odd peaks or it deviates from the line too much, then that indicates again maybe you've got problems with your data. The per base N content refers to how the machine assigns sequence reads. So if it's not confident in assigning one of the 4 bases, then it'll just call it N instead, basically meaning I don't know. So if you have good quality data, obviously you're not going to have very many N's. But in the bad quality data, there's a little area of the read where you can specifically see it's having trouble. So maybe this is a repetitive sequence or something like that. The sequence length distribution is good in both cases here, actually. So you'd expect all of the sequences to be the same length, which would be the length that you've programmed the machine to read. So actually in this case, all of the reads do seem to be the right length, but sometimes if you have particular errors, you might get reads that don't go all the way through, and so you get slightly smaller reads than you were expecting. Again, if your data is well balanced, then you won't duplicate too many sequences. But if you start having some contamination or perhaps very repetitive short sequences in your data, you might get areas where the same sequence is sequenced more times than on average, and so that shows up in these peaks in this graph. And again, it'll look through and identify specific sequences that really show up a lot of times, and we'll run those against a database to try and identify. So this one says that it's identified it possibly as being contamination from one of the Illumina primer sequences. And finally, it'll have a look at the, again, against a database of adapter sequences and make sure that you don't have any contamination from the adapter sequences. And the only sequence you're looking at is the actual sample itself in the middle. I'm trying to change slides, but I'm very close to the end. Where's PowerPoint?
Not that one, that one up higher.
Okay, back to PowerPoint, and I'm just going to say a very little about sequence trimming and Trimmomatic. So another program which is very commonly used, either to remove these adapter sequences, which we've added earlier on during the sample preparation, so that you only analyze this green section in the middle, or this is back to our bad quality data that I've just shown you. You might want to just trim off everything to the right of this, for example, and just keep this sequence for analysis so that you have only the good quality sequence to work with. So again, there's lots of different tools that you could use for this. Trimmomatic is one that we use a lot, though, and is very widely used. So it was developed to identify, solve 3 challenges: identifying adapter sequences within your data, maintaining the connection between paired end reads. So if you've read from both ends of one section of DNA, then you want to maintain that connection between the two, and being able to achieve all of this easily and quickly so that you're not spending a lot of time or a lot of money on doing this early stage of data analysis. Unlike FastQC, Trimmomatic can only be used in command line mode. And so you won't be able to sort of go on the internet and just upload samples, perhaps in a way that you might be more used to. But hopefully by the end of all of our demonstrations that we're going to go through, we'll have a reasonable idea of how to use command line tools. And we will use Trimmomatic in one of the upcoming tutorials. So if you're simply trimming adapter sequences off, you can just supply a list of the adapter sequences that you know that you've used, and the program will then identify those sequences where they exist and trim them off your samples automatically, or it can identify sequences that you might want to get rid of. So for example, if you have a very short insert length, you might find that the sequencing runs all the way through the data you're interested in and goes into the adapter sequence at the other end. So if you run it in what's called palindrome mode, the program will identify where this happens and trim off the adapter sequence for you. So I'll show you these slides later and you can have a look at the further information. I won't introduce it now. And again, I don't think there's any need to go into the summary. You can have a look at that again later if you want. I will just say there's a very useful playlist on YouTube, which I link to here, which you can watch in your own time. There's 19 videos, so it lasts for a long time. And it starts with the very basics, which we've been introducing here, and goes all the way through to some quite high-level applications. So you'll probably find that useful to recap on a lot of what we're going to be going through in the next couple of days as well.